Good evening, everybody. Am I audible and is my screen visible to all of you? Thank you, sir. So good to see you here. All right, so we'll start without wasting any more time. So now this session for today and two more sessions which are coming up later are basically based on the demands that I got from various students that they wanted some sessions on automation. Because you usually get a question on automation in bacteriology, automated serological testing, automation in molecular testing. So based on that, we thought we'll cover the major portions in 3 sessions. So I'm going to talk about automation in bacteriology. And next week, we'll have Dr. Sanjeev talk about serology, and then Dr. Uwais again talk about automation in molecular. And he's going to start from the basics and then go on to the automated molecular testing. Dr. Sanjeev is a professor at Lady Harding and Dr. Oves is a professor at Maulana Azhar. So if you have any queries related to anything else, you can ping me, put it in the chat. If you have any queries or any questions, I'll try to answer them as much as possible. If not, we'll take it forward and we'll get back to you. So starting with automation in bacteriology, for many years, we believed that automation in bacteriology has no value. We've all seen lab medicine labs where there are combined biochemistry, pathology, and microbiology, and we've always said, no, 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 we can't automate microbiology. One, because we feel that it's too complex. The samples are very variable. Something comes in a liquid form, something comes in a solid forms. There are tissue biopsies, then there are swabs which come, and we really don't know how the machine is going to pick it up. So there are various methods of collection, and then there are various issues with collection itself which are going to impact our result. And then there are various SOPs for processing of various samples. So all these becomes too complex. And secondly, we believed in ourselves that we are, irreplaceable, and all the reporting needs trained eyes. And whenever we talk about microbiology, we feel that I have to see the colony to believe that it is there. How many colonies are there? How often do we get this particular organism from this sample. And the last one, but the most important one, is probably that there is the automation is underutilized. Because of these two factors, the automation is underutilized. And anything which is underutilized is not a very good business model for businesses. And that is why the cost remains high. If these were utilized just like, the sample processors in biochemistry, the total lab automations that they are moving on with robotic arms and everything, and even in the clinical hematology lab, I think the cost would have come down. So, and especially if you have small business where the number of microbiology samples are less, it's going to remain underutilized in those labs. And again, we will feel that we are paying a lot of money, but it's not going to work. So, those are the reasons why the automation has not kicked off as fast as it has kicked off for biochemistry and hematology. But there is always a need of automation. Our clinicians require faster results, especially for identification and AST, and with the rational antimicrobial prescription and with antimicrobial stewardship, it's very, very important that we go ahead with this. Then we all have realized that we face manpower crunch, not only in our offices, but also in our homes. We face a lot of manpower crunch now. There are lack of skilled people availability, and there is increasing volumes. Every microbiology lab, there's an article which I read which says that there is a 10 to 15% increase in every hospital. Now, those increasing volumes with shrinking manpower, shrinking resources becomes very difficult to process. All the labs are getting accredited, even the government ones. So we want a quality assured results where the errors are minimized. And there is availability of technology and money. Earlier, we used to say, even if we automate, who's going to see our result? Now, what has happened is that we know that even so as to say, even in the villages or even in the lowest possible areas, there are people who've got internet connectivity and who can download the reports in app. So what is so specific about bacteriology? Now, bacteriology, as I said, is a very complex game altogether because there are too many samples, too many types of samples, and too much of processing which is involved, as you can see here. Now, what happens is that there is a situation where there is a specimen processing. Now, do you need an automation at this level? Yes, you do. You need an automated inoculation and streaking system. You may need a barcode tracking and a standardized plating methods. Then when we talk of culture incubation and imaging, we just put the plates in the incubator, forget about them and read them the next day. Is it possible that I read them in a real time, just like we have a real time PCR, which has completely changed the game of molecular testing? Do we have a real time monitoring where we monitor that, okay, because the colony count was so high, this colony is already grown in 5 hours or 6 hours or 12 hours rather than 18 hours? Am I able to release that report at 18 hours? There is a preliminary colony recognition and the instrument has beeped. Can I now take out that plate and do some work on it? Maybe identify it. Now, this colony selection and identification MALDI has been a game changer here, an automated system. Those institutes which have MALDI have seen it, how readily it changes the identification protocols. Similarly, fully automated systems which incorporate all these things together, colonies, antimicrobial susceptibility testings, and are fully integrated with LIS, also work very well. Now, so you may have an automation which is limited, say, only to the inoculation part. You may have an automation which is a partial lab automation where you have an inoculation part plus also a smart incubator, something like a VASP lab. I'll cover to this in the last part of my talk. And then you have a complete lab automation where you actually put it, shut it, and forget it till the report is ready, until you have an intervention only at the last end where you want to find out as to what is grown in which sample. So you will have different kind of systems which are available for specimen processing. You have those which are incubators and digital imagings where you have VASP labs or WCS or bench incubators with automated cameras which take images and then read these images with telebacteriology. I can sit at my home And actually, the reality of work from home can be there for a microbiologist as well. And then the follow-up work, the plates, the delivery, and other things which can be done by an automated system. So, what is the availability that we have right now? We are all moving towards the manual only, but what all we can automate is what is shown here in a orange colored thing. So, what do we talk about the specimen processing? Here you have something which is called as an isoplater. This came in 1989. It was the first semi-automated plating instrument. I don't know how many we have these in India, but I saw this in work in Karolinska Institute in uh, Stockholm when I went there in 2012. And I found that it just where goes a robotic arm goes, opens the cap, puts the loop inside, takes it out, puts it on the plate, rotates it, and then stacks it, and then sends it to a tray, and the tray directly goes into the incubator or a conveyor belt. So they had this semi-automated plating kind of an equipment. Later on came Innova from BD, Inocula A, which is part of the total lab automation system of BD Kystra, and a walkaway specimen processor, something like a walkaway biochemical analyzer, which is also called as the WASP, which comes from Copen Diagnostics. And then we have a Privia Isola, which is a bioMerio instrument. So what the isoplater was doing, isoplater was only, you label the plate, keep the label away from the edge of the plate, and keep that plate away from the frost inoculation mark, because that is the mark which the isoplater uses to locate the start position. And you put the petri dish media side down in the load carousel, and then you connect it to your samples. So it will create a barcode, put a barcode, and do it. The isoplater was not able to create the barcode. Now systems do. In Privia Isola, again, it's a patented robotic system which manages 90% of the steps which are required. You can see here there are various types of culture media that you can put. If you want, now there are those which have 2 heads, 3 heads. Suppose you want for each sample one blood agar and one McConkey. So you can put them separately here also, or you can put them one by one also. So agar plate with well separated colonies, eliminating unnecessary subculture, which provide results within 24 to 48 hours. Very often we are stuck with a situation where the culturing is not so good, and the end result is that we waste 2 days subculturing the organism in pure culture before we put it up for MALDI or for AST or for white tag and so on and so forth. So similarly, Kystra Inocula automated plating system, it's got a media storage and plate loading. It's got an automated barcoding and a fully automated robotic arm processing, like we see in the automated RNA extractors or DNA extractors that we have in our molecular labs. Then it has a biosafety cabinet where we do a semi-automated processing. Why semi-automated? Because here you have to load the sample with your own hand. Now there is another one which is WASP, which I said as you fill it and shut it and forget it. It also does an automated streaking, and in addition, it can also do a grams slide preparation from the plate which tests positive. So what are the features? The method of inoculation in WASP is a loop based. You can change the size of the loop if you want. Whereas in Inocula, it's a pipette based system. Streaking method is a rolling bead, whereas in a WASP, it's a L-shaped spreader, which does that. The wastage will be for bead as well as for pipette, and here it will be for reusable lamp. Both of these have got an automated decapping and recapping, so that the discard is also capped when the machine discards the sample. As you can see in Inocula, up to 12 media, different type of media can be inoculated for a simple sample, whereas for WASP only 9. I think this is also a very high number. Inocula can cater up to 270 samples in one go, whereas WASP can cater up to 72. And similarly, the cost is also 3 times for Inocula. There is a continuous loading. There are 2 kinds of systems. In one, it is yes, and in another, it is no. The plates which are streaked, as much as 5 plates can be streaked in one go. The non-liquid samples nowadays can also be processed in this, and there are optional modules where you can have an automatic gram slide preparation, and you also have a broth inoculation where you can, instead of the solid media, you can also put your tubes which are there. For Inocula A, which is an open platform, you can use any tubes, whereas for WASP, it has its own tube which are called the Copen tubes, 5 and 10. Again, it's a optional mode, but for certain samples like CSF, pleural fluid, you may want to inoculate a broth culture and it works very well for that. Then there are blood and fluid culture system, which not only take blood, but also take sterile fluids like CSF, pleural fluid, or any other body fluid which you consider as sterile. Now we are all familiar and we've all grown up with these systems, the bacteria alert system or the BD backtack system. One, either they are based on changes in the carbon dioxide or they are based on the changes in the fluorescence. Then there is a VersaTrack, a very similar system to Bacteria Alert from Thermo Scientific. There is an Autobio BC120, which uses a temperature control and a vibrating module to shorten the detection time. And then you have a Labtronics, which is a fully automated system with an optical algorithm that tracks fluorescence. Then you have for Mycobacterium tuberculosis, you have midget, which again is a very familiar terminology. So I'm just going to talk a little bit about them as to how they work. Now, on one side is your bacteria alert, that is on my left hand side, and then my right hand side is your BACTEC fluorescent test technology. So, what happens in bacteria alert is that the carbon dioxide, which is released by the bacterial metabolism, it permeates the LES, which is fixed at the bottom of the bottle. Now, each bottle contains certain compounds. It contains the media, it contains certain resins. We all know that. It's a basic undergraduate that what does the bottle contain. But what it does here is that this LES, the carbon dioxide, combines with the water there and forms carbonic acid or H2CO3. This is going to cause a fall in the pH and is going to increase in reflectance as the pH falls. Now, as the pH falls and the growth curve enters in the log phase, the black bottle is flagged positive. Now, in the BD bottle, something little different happens. What happens is that the organism metabolic activity again releases carbon dioxide, which will act with the dye which is there at the bottom. And this LED which is there gets activated and it activates a fluorescent material which is there in the sensor. Now, this fluorescence is modulated by the dye in the sensor and the photodetector will then detect the fluorescence. And this will then give you a positive analysis and which will then be indicated in multiple ways. Either it will give you an alarm or it will give you a light or LED. Can everybody please mute themselves? All right, so this is what about this. Now, how does the midget work? Now, midget, we all know, is comprised of a media called the modified Middlebrook 7H9, which is 7ml, and it contains growth supplement, which is essential for growth of MTB. Now, it also has certain antibiotic mix, which prevents the contamination, and it has an oxygen quenched fluorochrome, which is embedded in silicon at the bottom of the tube. Now, here, because it's a slow growing organism, again, the purpose is same, that the free oxygen gets utilized and gets replaced with the carbon dioxide. As the oxygen depletes, the fluorochrome is no longer inhibited. Because you see here, this is an oxygen quenched fluorochrome. As the oxygen concentration goes down, this fluorochrome is no longer quenched and the fluorescence of intensity will be proportional to the extent to which the oxygen is going down and will be increasingly proportional to the growth of mycobacteria. So again, it works on a fluorescence principle, but it is the oxygen which is going down, resulting in the fluorochrome which is no longer inhibited. So, like you can see here in a negative culture, there is a lot of carbon dioxide which is happening and the sensor has got an FO2. So there is little or no fluorescence. But as the organism grows, the oxygen gets utilized and the quencher gets stopped and you get a very strong fluorescence here. Now let's come to automation at the level of culture incubation and imaging, something which sounds very, very dramatic and something which most of us are yet to see, but I'm sure the younger generation, this is a PG Masterclass, so all the PGs here will see these very soon in the next coming few years in your laboratories. It not only automates incubation, but also continuously it digitally monitors the incubation. So it's got high quality cameras which have customized optics and plate orientation so that it continues to take images and tries to analyze them. Similarly, a Copen VASP has also got an AI driven image analysis and flags positive culture. So what it does is that there are culture plate images for initial review. Then at the reading stage, they are also displayed for detailed examination. And the selecting stage, you can create an algorithm where the selected plates can directly go for workup. You can attach a gram stain module where the gram staining of these plates can be done automatically. And the software can then send a result to LIS and also archive these cultural images as to what is happening. Now, something which at an incubation stage, which is there, but is a smaller one, is a Symbiosis Ecolite 3 system, which has a lot of, then there are protocol system and a Unistat, which is a statistical analysis module. What it does is that it is a quality counter. So what you can do is maybe after say 12 hours, you have to manually put the colonies here and it enumerates them and it analyzes them and it also gives you the result. So it also can read your antibiotic sensitivity plate. It can also read your maybe urine culture plates and give you a result. Now, the whole problem with this automation is that for one sample, you will require one plate. Normally, we are used to culturing 2 or 3 samples on one plate. But that is not going to work here. You will require us one plate for. It can be customized to use smaller plates, but that is what it will be another burden on the lab. Now, at the stage of identification of and AST, we all know about certain automated systems. There are 3 or 4 which are available. First is your Vitek 2 from bioMiro, which works on an automated bacterial yeast identification. It uses a combination of calorimetric and fluorimetric technology. It can give rapid ID in 2 to 8 hours and AST in 6 to 18 hours. It's got a broad organism database and a resistant system. On the other hand, there is BD Phoenix from BD Diagnostics, which uses a redox and a turbidometric analysis, and it has an advanced expert system to interpret resistance mechanisms. Now, how does the Vitek work? Vitek works with the fastest this thing and is one of the most efficient flow. So, but what is the technology on which it works? So, if you look at the identification, we all know these Vitek cards. These are 64 wells with an individual test substrate. Just like we put up the biochemical test, similarly we put up these cards which will have substrates which have for acidification, alkalization, enzyme hydrolysis, and growth in the presence of inhibitory substance, just like we have urease, citrate, all these. And then there is an optical clear film, which is there on both sides, which allows the appropriate level of oxygen transmission while maintaining a sealed tube. And it has a pre-inserted transfer tube, which is used for inoculation. So how does it work? It works by reading these systems in an optical system at these wavelengths. So it identifies the organism on the basis of same phenotypic results. But because these are micro quantities, the results are produced in a very short time. And then the software is built in such a manner that it is able to analyze this raw data and give you a first level algorithm. Now, this first level algorithm will stop unless and until it is able to match with an organism. If it is not able to match this kinetic analysis, then it will continue to read further and detect these changes and further go ahead. So it will transform a set of successive reading into a test which is plus minus, like we do in our lab. What we do, like in API strip, what do? We read urease positive, so we write plus, citrate positive with growth, so we write plus, TSI with H2S, K by A, so we write that result. So immediately it converts. Now, then it starts converting into a bio number, and then the algorithm will convert this bio number into an organism. So for each well which is there will be 3 reading, and there will be 16 steps. Similarly, AST is again a miniaturized version of broth microdilution MIC technique. Now, it is based on MIC technique. It does not mean that it is giving you a true MIC. The system actually what it does, it monitors the bacterial growth in these wells. You can see there are 64 wells and then there are 16 antibiotics. So you cannot have more than 4 wells for each antibiotic. That means you can't have more than 4 dilutions. So what does it measure? If the organism is growing in the presence of that well, it means that it is measuring the turbidity. And then it is using that data to tell us whether at this concentration the organism is there or not. That is your principle of MIC. That is also the principle of Vitek. But what is different here is that when we talk about this time to maximum growth and we look under the area of curve as to what has happened here, then the algorithm is built in such a manner that it will compare it with an organism which has a known MIC. Suppose the MIC of an organism was 2, what would have happened? Does it match with 2? Does it match with 4? Does this growth kinetics and this area under the curve match with an MIC 8? And that is what the algorithm does. So this, what is the length of this phase? How fast is it increasing? What is the area under the curve? All these are built into the system where the system will. Now, to see this in a further manner, the organism activity, suppose the MIC reference value is 0.25. 0.25 is something which is, say, that it is a very, very sensitive organism. So this will be the curve, which is similar to as if there is no antibiotic which is present in the Vitek 2 well. So this blue thing is actually your control. Now, if your reference MIC is 1, so what will happen? This green line will tell you that there is an MIC curve, which is how is it different from the organism. Then when your MIC is say 8, let's come to this, then what's happening? So you have different versions here as to what will happen when you have 6 hours, 9 hours, 12 hours, 15 hours, and 18 hours. So if the red line here will tell you when the antibiotic concentration is actually the 4th one, that is your, say, 2. So if you had 4 concentrations, 0.25, 1, 2, and 4, this is how your curves will look like. Now, in order to, it will become very confusing if you see all these curves and then make a decision. So what it does is that it plots it into a 3-dimensional curve. So each axis plots relative organism as a percentage as to how many you got which would have matched above 8, above 4, and above this. And then it calculates the MIC from this plot. Now, the other system is a BD Phoenix system, which works both on a redox indicator as well as turbidity growth detection. So it utilizes a panel where there are doubling dilutions. So here the panels are slightly more. It has about 136 well, and it also has a combination panel. This does not exist with Vitek. ID side has 51 wells and the AST side has 85 wells. So you see it has a little more dilutions which are available to you for calculation of MIC. So in Vitek 2, it is a computer-assisted extrapolation of doubling dilutions. Your MIC may be between 2 and 4, but the computer will extrapolate that depending on the growth kinetics, it is matching with 4 or it is matching with 2. Whereas the BD Phoenix system will give you that the antimicrobials are usually in a serial dilutions and they are based on slightly more dilutions than they are in the Vitek system. They are also not giving you an exact 16 dilution of MICs. That is not possible with 136 wells. Although most of the time companies will come to you and claim that we are doing true MICs, but they also they have slightly more dilutions. Then the Phoenix, as I said, depends on a redox indicator, whereas that it is only a growth indicator using a optical scanner, which I told you. Now there is a microscan walkaway ID and DST system, which has these panels with 96 wells. Now this system actually works on a direct MIC detection, which has multiple dilutions and does not rely on a historical data or a virtual MIC, because all the others are actually comparing it with an algorithm which has historical data. So it's not giving you a virtual MIC, it's actually giving you a true MIC. Now, when you talk of the why I have covered this slide is that don't just shut it and forget it. There are certain automated systems will also require a quality check, a purity check plate before and after inoculating your Vitek cards, density check plus calibration, your saline per lot QC. Whenever you get a new shipment, put up the standard controls. System performance QC. Very often, what happens is that the engineer will come and they will do the system performance QC, and I'm done away. I don't have to do anything else. But then there are daily purity checks, there are a daily calibration check, maintenance check, documentation check, and density check plus cleaning, weekly saline cultures for sterility. All that you have to do whenever you are using this. Now, coming on to another game changer, that is the MALDI-TOF. The MALDI-TOF actually stands for matrix assisted laser desorption ionization, where samples are actually fixed on a crystalline matrix and then they are bombarded by laser. So when they are bombarded by laser, this sample analyte actually evaporates. When it evaporates, it creates certain signatures. And then TOF stands for time of flight. Time of flight means that these ions are actually separated by their mass to charge ratio. So what will happen is the lighter the particle, the faster it will flow in this flight tube. The heavier the particle, it will flow slow. And depending on how it gives, it will give you certain amount of your mass to charge ratio will give you certain amount of, say, peak, which in the mass spectrometry will correspond to a mass of that particular particle. So this analyte, once it has gone through this, the detector will read it. And what it does is that not only does it read it once, it also reflects it so that they fly towards the second detector. This kind of extends their flight distance and also makes it a lot of focus. And then it combines this data from here and gives you those peaks which are there, which are this mark spectrum depending on how these mass to charge ratio particles have got vaporized is very, very specific signature for a particular organism. And the database will have all these and it will continue to give you this result. Now, what are the companies which are available? One is BioVario and the other is Bruker, which is available for this. There is not much difference between the two. Some give you filamentous fungi, some give you only bacteria, some give you bacteria plus yeast only. So this is all you require. But there are two things you should remember here. One is that the bacterial matrix is slightly different chemically from the fungal or the mycobacterial matrix. Understandably, because you need to break down the cell, and the cell wall of fungal and mycobacteria is slightly different from bacteria, and that is why this chemical is different. So you have an alpha cyano for hydrocinamic acid or CHCA. Along with that, you have a synapenic acid for fungal and mycobacteria, and whereas in you have acetonitrile and TFA, TCA, that is trifluoroacetic acid for bacteria. And formic acid extraction step may be required when you're using fungal. Now you may have Bruker biotype. Then there are, these two are available in India. The eczema, ceramans, andromas is not yet available in India. And each manufacturer has developed a peak matching algorithm to compare with a known spectrum. Now, what is the problem here in MALDI as well as in Vitek is it will only give you to a species level. For example, the problem comes with salmonella. It will only tell you that it is Salmonella enterica and not go any further. Whereas you require, whether it is typhi or paratyphi, for that you will need serotype. The other issue here, as well as for Vitek, as well as for MALDI, is the close association of Shigella with Citrobacter, Enterobacter, and other organisms, and with E. coli. There are certain difficulties in identifying Shigella when you look at these two systems, whether it is Vitek or it is. Now, what is the advantages of using these automated technologies in bacteriology? One, it significantly decreases the turnaround time. The sample preparation time is very limited. You can tell there is not much a very complicated requirement. So when we have the growth, even on the next day in the lab, we inoculate the MALDI-TOF. within a few hours, and we find that by say 11 o'clock or 12 o'clock, your report is ready. Then these are all very robust interlaboratory reproducibility is there. A lot of research has gone into it. These are cost effective. The running cost of multi is hardly any cost, it's less than maybe $1. But the initial machine is very expensive. And there are little adaptable systems. For example, today my lab takes only 50 cultures, so I can buy a smaller version of Backtech and tomorrow move on to it. It is expandable. You don't have to buy the new machine every time. What are the limitations? Limitations are that identifying a new isolate is possible only if the company decides to include it in their database, whether it is Vitech or it is Maldi. Then I already told you that there are certain organisms which cannot be identified. Uh, Shigella is one, pneumococci is another. Of course, salmonella, you have to go for serotyping. And this, as the coordinator for Delhi Network for AMR, this is often a problem that what we find as salmonella may not turn out to be salmonella when we do serotyping at our center. And it is not useful direct. Nothing is useful for direct testing of clinical specimens as of now, although there is a variation of MALDI where you can use from directly blood culture, flagged blood culture bottles. There is a system to extract centrifuge, a specific centrifuge where you extract the material and you can use it for MALDI. Now, what are the total lab solutions which are available? There are currently 3 total lab solutions which are available. One is FMLA, then Kystra, which I've already talked about, and the WASP lab. These have got robotic arms, conveyor track systems, they capture the images, they have a computer-driven robotic plate management. And initially, I told you what are the difference in their system. Here is about what are the loading, how. So they can handle a really high load, 600 plates an hour or 360 plates an hour. So it's very good if you're a referral center for a big city where all the small lab or collection centers are going to send you the samples. Imaging time is also specified. Definition of the camera is very good. And the size is as high as about 3 MBs. So what are the basic advantages of automating? We talked about that there is a need of automation. There are certain hinges which are there when we talk about then what is the need to automate? One is activity and productivity. There is an improvement in the workflow. There are management of reports and there are a lot of cost savings at the end because you save on excessive manpower, you save on repeat subculture, you save on repeat ASTs. Quality and reproducibility, yes. Inoculation, definitely yes. There are very less chance of the contaminated cultures. Growth is improved and there is a reduced time to result. Decrease hospitalized time and then indirect cost savings. If your patient is hospitalized less even by 48 hours, you save on a lot of money on maybe the workforce, the medicines, and also on healthcare associated infections. Disadvantages are that you cannot say that I will work with an automation and not have a lab which runs for 24/7. Because if something is going to beep at 3 o'clock in the night, somebody has to be there to take care of it. Secondly, these tools can be misused. Third, expectations for increased productivity is not achieved. If there is no 24/7 lab, the owner of your hospital will say, why do you want an automated system when you can't give me a report at this? And our samples have not increased even after spending money on automation. Then you need a backup. Today, we have faced this in our lab also. We do most of our work on Maldi. And sometimes when Maldi breaks down, it becomes really difficult for people to actually go and work on traditional systems. So you need a good support and maintenance system and an expensive maintenance budget. You can't afford for the machine. Then the staff escapes, if there are 600 plates which are there, the staff is only going to monitor that machine and sit with their arms closed, that there is nothing to be done. And slowly the technical knowledge is being lost. I often tell this to my students in the lab. You are forgetting how to inoculate and read a TSI because there is a MALDI in the department. And the conventional workflow, people like me and any other senior that you talk to, they will feel that, okay, I know this by my hand and I have a pride and an ownership. So that pride and that ownership that this is my lab and this is how I work can also go down if machine is doing everything. And then of course there are specific disadvantages which are limited to a system. The photo capturing may not be good, you may not be able to read it properly, the organisms, certain organisms are not identified properly, and all these systems will be there. So thank you so much. I think with that I will end my talk. If there are any questions, please put it up in the chat box.